Hey, this is the second lecture video for the genetics module. So in the first lecture video, I went over an overview of genetics and defined some terms for you, such as allele, homozygous, heterozygous, dominant, and recessive. So now you are going to apply that information and learn about Punnett squares and Mendel's law of segregation and law of independent assortment. Now here it says pedigrees and inherited diseases as a topic. That will be the third lecture video for this module. So this is the slide that I stopped at in the previous lecture video. Now I mentioned before that Gregor Mendel is the father of genetics. He was a monk that lived in Austria. And he did many, many, many experiments looking at inheritance of traits between parents that are reproducing sexually and offspring. Here are a few more definitions or terms that you need to know to understand Mendel's experiments and genetic crosses. So here is what we call the parental generation. So this is the original cross when you are doing genetics experiments. Now, the organisms in the parental generation reproduce sexually, and the offspring are called the F1 generation. So the parental would be like your parents, the F1 would be you, and then the F1 reproduces to give you another set of offspring that we call the F2. Now that would be like your kids. So it would go parents, you, your kids. So 3 generations. Now, when we do genetics experiments, we call it the P or parental generation, the first generation or F1, and the second generation or F2. So now I am going to give you a few more terms. Okay, yep, still have a line left here. Okay, this is a little blurry, this image, and I apologize about that. I did not know it would be blurry. So we were talking about flower color in pea plants. I'm going to look at seed color for this example because I liked the way that they laid it out in this image. So here we have the parental generation. You see that P right there. Now, what I would like to point out here is that in the parental generation, so we're looking at seed color, it can be yellow or green. So one of the plants had yellow seeds, one of the plants had green seeds, but notice that they are both homozygous. So the yellow seed plant is big Y, and the green seed plant is little y, little y. These are what we call true breeding because they are homozygous. So homozygous individuals are true breeding individuals. Okay, move my cursor here. So true breeding refers to a homozygous individual for a particular trait or gene. Now in Mendel's experiments, he always started with true breeding plants in the parental generation. So now what I'm going to do is go to the whiteboard to work out this cross. So we are going to talk about seed color. So first of all, remember that there are two alleles for every gene, at least two alleles. So for this example, we are looking at the seed color gene. Big Y equals yellow, little y equals green. Now remember that we give the capital letter to the dominant allele, the lowercase to the recessive allele. Okay, so we are crossing big Y with little y, little y. So this is our yellow seed individual. And this is the plant with green seeds. So the point of Mendel's experiment were to understand how alleles, or really traits, are passed from parent to offspring. He had to actually do these experiments before he thought of the idea of alleles, right? So he did not know about alleles when he started. These were the experiments that taught him about alleles. Okay, so this is our parental generation. I'm going to put P right here. That actually may be confusing. So once you set up the cross, you need to determine the gametes for each plant. Now, this is going to go back to meiosis, and this is why we did meiosis prior to genetics. So this is sexual reproduction. And so we are looking at egg and sperm. Fertilization produces the offspring. Now remember that the egg and sperm are haploid, half the genetic information. So one chromosome per pair, okay? Now, during meiosis, if big Y were the alleles on the homologous chromosomes, those are being separated, right? They go to the gametes. So in the yellow seed individual, let me see, one of the gametes would be big Y and the other gamete would be big Y, right? They're being separated, but they're the same allele. They're both big Y, which means we'll just say the yellow seeded plant is donating the eggs. So what that means is every egg produced by this plant will have big Y. There's no other option. So the gametes from big Y would be big Y or big Y. Now, as you probably can guess, the gametes for the green seed plant are little y or little y. Now, it is important that you determine the gametes as your second step here, right? And this is on our way to creating a Punnett square, because you are going to put the gametes across the top and side of your Punnett square. So let's take a look at that. Now, I want to emphasize here that a Punnett square, we do Punnett squares to determine the probability of getting offspring with a certain genotype and phenotype. So I'm going to write that down for you. Let me just finish my Punnett square here. So Punnett squares are used to determine the probability of an offspring having a specific genotype or phenotype and or phenotype. So we're looking at probabilities, right? The Punnett square is not telling you that there were 4 offspring. It's probabilities. There could be 200 offspring. There could be 10 offspring. Okay, this is not the number of offspring. So I'm going to put my yellow seed plant across the top, but it doesn't matter if you do the yellow seed plant across the top or the green seed. I just always put like whatever individual I put first in the parental cross is the one I put across the top of the Punnett square. But like I said, it doesn't really matter. Okay, down the side, we will put the gametes for the green seed plant. Now notice that I am only putting a single Y per box, right? A single Y for the box on top, single Y for the other box, and then single Ys down the side. I'm not putting big Y for one box because these represent the gametes. So across the top, these would be like the eggs from yellow seed plant. Okay. On the side, this would be the sperm from green seed plant. Now it doesn't quite work this way, but our purposes, it does. Okay, there we go. Okay, now all you have to do is fill in the boxes. Okay, the boxes will be the offspring. They have homologous chromosomes, so you're joining those gametes in fertilization. So let's look at the first box here. You bring that big Y down, that little Y over, and the genotype is big Y, little y, okay? So in the same way, you bring this gamete down, and then this gamete over, and you get big Y, little y. Now, just a note here, I always write the capital allele first. If you get used to looking at it that way, you're not going to overlook like a genotype or, like look at it wrong, I guess, when you're determining phenotype. So now this box, we have big Y, little y, right? We have a big Y on top. For that column, you bring it down, you have a little y on the side. And same for this box. Okay, so now I have filled in the Punnett square. So let's look at what we do with this. So first of all, this is our F1 generation. And I want to restate the fact that I am not saying there are 4 offspring. Okay, well, let's say there are 100 offspring. I'm gonna write that right here. Okay, we are going to say that there are 100 offspring produced from the parental cross. So now we are going to look at the probability of obtaining a certain genotype. So what you do for this is you look in your boxes and you look at the different genotypes, like how many different genotypes did you get and what are they? So for this Punnett square, we only got one genotype, big Y, little y, okay? Now, we're talking probabilities here. So what you do is you count the number of squares that have big Y, little y. And that will be your numerator or the top of your fraction. The denominator or bottom is the total number of squares, which is 4. So 4 of our squares have big Y, little y out of 4 squares total. This equals 1, which equals 100%. Little refresher on percentages. Once you divide your numerator by your denominator, right, top by bottom, you get a number. To get the percentage, you move 2 decimal places to the right. Okay, so remember the genotype, you are looking at the specific letters, the alleles that are present in the individuals. So phenotype Think phenotype, physical. So it's physical appearance. Now, if you look over to the upper left-hand corner, I told you that yellow was dominant because I gave it the capital Y. So let's look at our genotype, big Y, little y. That means that all the offspring are yellow. If there is a capital Y at all, the plant has yellow seeds. Okay, so the only phenotype present is yellow seeds. 4 out of 4 squares have yellow seeds, which equals 1, which equals 100%. So remember, I told you there were 100 offspring in the F1 generation. So 100% of those offspring have big Y, little y, and 100% of the offspring are yellow seeds. Okay? That was our F1. Now, Mendel always looked at the F2 generation. So technically, he let the F1 offspring self-pollinate, which means that you had a big Y, little y times big Y, little y. We have big Y, little y, and big Y, little y. So this is the F1 that are reproducing. We have a yellow seed plant and a yellow seed plant. Now, when Mendel was originally doing this experiment, he didn't really understand alleles or dominant or recessive, but the fact that he could take a yellow seed and a green seed plant, put them together, and all the offspring were yellow, told him that somehow the yellow trait masked the green one. So that's what started to give him the idea of dominant versus recessive versions of a gene. Okay, so now we are going to look at the gametes. So remember, the homologous chromosomes are going to separate into 2 different haploid gametes. So the gametes for the first individual could be big Y or little y. And same for the second individual. So now we are going to draw the Punnett square. Okay, and we will put the gametes from the first individual on top. We have a big Y and a little y. And I actually recommend you pause the video briefly and try to do this on your own to kind of check your understanding. Now we have a big Y and we have a little y. So just to remind you, we will say that these are the different eggs and these are the different sperm that could be created through meiosis. So in this square, you have big Y, big Y. This square, big Y, little y. This square, big Y, little y. And this square, little y, little y. So all I'm doing is taking the gamete for the column and row and putting them together. So again, we are going to say that the F2 generation had 100 offspring. I'm just going to write has 100 offspring. So let's look at genotype. So look in the Punnett square right inside at the different types of offspring you can get. And you should see that there are 3 different genotypes. big Y, big Y, big Y, little y, little y, little y. So now we have to determine the probability of getting a certain type of genotype in the offspring. Now we express this as a percentage or a ratio. So let's look at big Y. 1 out of 4 squares has big Y. So that equals 0.25 or 25%. Whoops, percent. Okay, we have big Y, little y, and you find that in 2 out of 4 squares, which equals 0.50, which equals 50%. Remember, to get my number there, I take 2 divided by 4. Little y, 1 out of 4 squares equals 0.25, which is 25%. Now, for our ratio, I would say that the genotypic ratio is 1 to 2 to 1. So I'm just looking at the numerators or top of the fractions. Big Y is 1, big Y, little y 2, little y 1. Okay. So in terms of number of offspring, 25 of the offspring should be big Y, 50 of the offspring, big Y, little y, 25 of the offspring, little y. Now, it's not going to be exact, right? This is just like a range. So like instead of 25 big Y offspring, you could have 28, you could have 23, it varies, but you have to be in like the ballpark, right? So now let's look at phenotype. We have yellow in our squares and green. Remember, phenotype, physical appearance. Now, if an individual has a capital Y in the genotype, it is going to be yellow because the yellow allele is dominant. It covers up or hides the green allele. So let's take a look at this. Big Y and big Y, little y are yellow. So we have 3 out of 4 of our squares are yellow. Little y is green. So 1 out of 4 squares is green. Remember, the recessive allele only shows up in the phenotype in a homozygous recessive individual. So 75% of the offspring should be yellow, and 25 of the offspring would be green. Okay. Now remember that the phenotypic ratio, yellow and green, your yellows can be big Y, or big Y, little y. So if you look at the genotype that I wrote up there, 50% plus 25% means that you get 75% for that phenotype. Now the phenotypic ratio is 3 to 1. Okay, so Mendel realized that, okay, he had a parental cross, you have true breeding individuals, he got all yellow for the F1, meaning yellow is dominant. But to his surprise, in the F2, that green seed showed up again. Now that clued Mendel into the fact that alleles exist. So let's go back and look at our slide. Okay. So this image is showing you exactly what we just did with the Punnett square. We have our parental generation, big Y, little y. We have the F1. All of the F1 are yellow with big Y, little y. They are heterozygous for the seed color gene. So we crossed a big Y, little y with a big Y, little y. Here's the Punnett for the F2. And the result is a yellow phenotype. Actually, let me do this. A yellow phenotype, and the ratio is 3. A green phenotype, the ratio is 1, so 3 to 1. And then if you look at the genotypes, big Y, big Y, big Y, little y, and little y in a 1 to 2 to 1 ratio. So you need to remember that if you start with true breeding individuals, the F1 has the same genotype, same phenotype, 100% big Y, little y, 100% yellow. You cross those individuals, and in your F2, you have a 1 to 2 to 1 genotypic ratio and a 3 to 1 phenotypic ratio. Okay, this is showing you the exact same thing, only it's looking at flower color in pea plants. Now, this is starting with big B little b times big B little b. So it's kind of like starting from the F1 in the example that I just showed you. They're calling it the parental. So in the offspring from big B little b times big B little b, you get the 1 to 2 to 1 genotypic ratio and the 3 to 1 phenotypic ratio. So exact same thing. 75% purple, 25% white. Purple being your dominant allele. So now we're going to look at the principles that Mendel put together based on his work. And the first one is called the law of segregation. It involves 4 statements. I'm going to read these and then I will write kind of like a simplified form that's a little more direct. So for the first statement, it says there are alternative versions of a gene that account for variation in inherited characteristics. So all he's saying here is that there are alleles and that these alleles lead to a variation in a trait. So alleles lead to variation in a trait. So our trait is flower color. The variation would be purple versus white. So I'm going to write that here. Okay, so that is the first, we call it a postulate or I guess statement that Mendel came up with. Now, number 2 says that for each inherited characteristic, an organism gets 2 alleles, 1 from each parent. So what this is telling you is that basically that the alleles for a gene or a trait, 1 comes from mom, 1 comes from dad. So in this case, the trait is flower color. So 1 allele for a trait comes from mom, 1 allele comes from dad. Okay, so far, Mendel said, okay, there's alleles, different versions of a gene, and these lead to variation in a trait, purple versus white. He also said that 1 of the alleles comes from your mom, 1 comes from your dad. And the other way to think of that is 1 comes from the egg, 1 from the sperm. If you're not used to thinking of plants as like mom and dad, which actually we don't refer to plants like that, but So there are two more statements that go with the law of segregation. Now remember, to segregate means to separate, right? So law of segregation is really looking at alleles being separated. So the third statement here, if two alleles of an inherited pair differ, then one determines the organism's appearance and the other has no effect. So this is just talking about dominant versus recessive alleles. So in a heterozygote, that's what he means by if the alleles differ. So in a heterozygote, one allele determines appearance or phenotype. And this is what we refer to as the dominant allele, even though Mendel didn't put that in his law. So one allele determines appearance, the other allele has no effect on appearance. That is what we call our recessive allele. Okay, so here Mendel got the idea of, okay, one allele is dominant, one is recessive. So if the dominant allele is present, then the appearance of the individual reflects that dominant allele. Just like we saw with the yellow seed, if it's capital Y, or capital Y lowercase, the seeds are yellow. So finally, a sperm or egg carries only one allele for each inherited character because they separate during meiosis. Now, Mendel did not come up with the term meiosis. He didn't really know about meiosis, but we know now that this is how this happens. So sperm and egg carry one allele for a specific gene. The alleles are separated in the sex cell during meiosis. You have a sex cell in the ovaries of a woman, undergoes meiosis, turns into a haploid gamete. Okay, the original sex cell is diploid. The gametes are haploid. So now we are going to look at Mendel's law of independent assortment and dihybrid crosses. So the cross that we just did is called monohybrid, mono meaning one. We were looking at one gene. Dihybrid cross, di means two, we are looking at two genes. So what is a dihybrid cross? So a dihybrid cross is a genetic cross that focuses on two genes rather than one. So in the cross you're looking at here, the genes would be seed color and seed texture. So I'm going to write that up here. So for this cross, gene 1 is seed color, and you have yellow or green. So for seed color, there are two alleles. Big Y equals yellow, little y equals green. Okay. Now gene 2 is seed texture. So in my picture here, they used R, the letter R for seed texture. So big R equals smooth, little r equals wrinkled. So how did I know that? Well, if you look at the picture, right, this first plant is yellow and smooth for the seed. And the alleles are big Y, and big R. We always give the capital to the dominant allele. Plus, if you look at the F1 generation here, you will see that all the individuals are yellow and smooth. That tells you that yellow and smooth are dominant. Now, in this second seed here, you notice that it is green and wrinkled. It's little y, little r, little r. Now, neither green or wrinkled shows up in the F1 heterozygote, meaning they're recessive. The only way you will have a green seed is if you have little y, little y. The only way you will have a wrinkled seed is if it is little r, little r. So we have the parental cross, true breeding, they're homozygous for both genes. Now, when I say homozygous or heterozygous, you have to talk about both genes, okay? Because they could be homozygous for seed color, heterozygous for seed texture. Okay, so he did the parental cross. I say he, meaning Mendel. All of the F1s were yellow smooth. They were heterozygous for both genes. We have big Y, big R, little r. Okay. Now, if you look at the Punnett square, it's not a 4x4 Punnett square because there are 4 different types of gametes that you can get from big Y, big R, little r. Now, what I want you to notice here, first of all, you do not have to do one of these problems in a quiz or anything. I just want you to be familiar with the fact that dihybrid looks at 2 genes. So let's look at the gametes on our Punnett square. Here they are, big Y, big R, little y, big R, big Y, little r, little y, little r. Now notice that in each gamete, there is 1 allele for seed color, 1 allele for seed texture. Okay, you do not put 2 alleles for seed color in one gamete and 2 alleles for seed texture in another. Each gamete has to have a chromosome or an allele for each gene. Now, the reason there are 4 different types of gametes is you have to look at every possible combination of these 2 genes. Okay, so down here on the left side, it is the same alleles because it was the same type of plant, yellow smooth. So then you combine these gametes and you end up with offspring with the full set of chromosomes for those 2 genes. Right? It is restoring the diploid state. So for each gene, it has homologous chromosomes. Okay. So that is basically what I need you to know about a dihybrid cross, is looking at 2 different genes. And you should be able to really see the process of meiosis here. Okay, those alleles for each gene are being separated and sorted into gametes. So now let's look at Mendel's law of independent assortment. So basically, Mendel figured out that each pair of alleles segregates or separates independently of other pairs of alleles during gamete formation. So let me show you what that means. Basically, he is just referring to crossing over. So let's look at a homologous pair of chromosomes here. So we will say that up here is seed color, down here is seed texture. Now remember that these are heterozygous individuals. So here we have a big Y, little y, and big R, little r. So let me write the genotype, the full genotype of this individual. This individual would be big Y, big R, little r. And they would be yellow, smooth. Okay. So the law of independent assortment means that crossing over can occur between the location of the seed color gene and the location of the seed texture gene. So let me write that down for you, and then I will kind of show you what I mean here. So independent assortment. So wait, I'm going to write a couple things here. So it refers to crossing over during meiosis. I want you to think a second and try to remember during which stage of meiosis do you have crossing over. I'm not going to tell you the answer. If you don't know, go back to the other presentation and look at it, like the PowerPoint. So refers to crossing over during meiosis. So crossing over, which is the exchange of alleles, occurs in between the locus for seed color and the locus for seed texture. Remember, the locus is the location of the gene on the chromosome. So here we have our homologous chromosomes. They're going through meiosis. So crossing over could occur right here to give a novel combination of alleles. And what this means is capital Y and capital R do not have to stay together on the chromosome. Little y and little r do not have to stay together on the chromosome. So these are homologous. During crossing over, say the y's could be swapped. So now you could have little y, big R, big Y, little r. So it would look like this. So this is after crossing over. So now we could have little y, big R, big Y, little r. So what that means is in the offspring of this cross, you could see like a yellow wrinkled seed or a green smooth seed, right? Yellow does not always go with smooth. Green does not always go with wrinkled. You have new combinations of alleles because of independent assortment, right? They're not stuck together. There could be crossing over between the loci. So independent assortment can give you new combinations of traits. So we could start with yellow smooth, just like the parental, but we could also have yellow wrinkled because of crossing over. We could have started with green wrinkled, like the parental, like the offspring could look like the parental, or it could be like a new combination, green smooth. So independent assortment means that when you are looking at more than one gene, that the alleles for those two genes can be swapped independently. Okay, they're not stuck together. Oh, wait. Let's see what I want to do here. So if we go back and look at this picture there, and you look at phenotype here, right here, you can see that there are different types of seeds: yellow smooth, green smooth, yellow wrinkled, green wrinkled. Okay, they are not all yellow smooth or green wrinkled. You have new combinations. That's what independent assortment results in. So just to summarize, you need to know all those terms I gave you in the previous recording and on this PowerPoint. You need to understand Mendel's law of segregation, what it says and what it means. You need to know how to do a Punnett square and get genotypic and phenotypic ratios. You need to know the difference between a monohybrid and dihybrid cross and what the law of independent assortment means. So as the last part of this lecture video, I am going to give you a problem to do. So hold on. Okay. And I'll work this problem out with you. That didn't work. Hold on 1 second. This did not share correctly. Why? Let's see here. Okay, I think it shared correctly this time. Okay, so this is like what you'll see on your genetics quiz. So in this problem, I am asking you for the genotypic ratio for the following cross. Now, I would pause the video and try to do this on your own. But for this video, I'm just going to continue to the solution. All right, so we have a big P, little p individual. You don't need to know what gene we're looking at. You should be able to figure it out without that information. And little p, little p. So first we're going to get our gametes. This individual can have a big P or little p. This individual is homozygous, so there's only one type of gamete, little p or little p. Now we're going to do the Punnett square. I recommend you always do a Punnett square. Some people are just super quick at this stuff and they just do it in their heads. That is not me. Whoops, I don't like that line. I mean, some things I can do in my head just because I have them memorized, right? Like a true breeding parental cross always gives you 100% genotypic phenotypic ratio. But like I said, I recommend you always do it. Write it up. So I'm going to put my first individual on the top, big P, little p. Those are my gametes. The second individual down the side, and that individual has little p or little p. Okay, now we just combine gametes, represents fertilization. So in this box, I have big P, little p. This box, I have little p, little p. Here I have big P, little p, and little p, little p. So let's look at our genotypic ratio. So look at the Punnett square and how many genotypes do you see? Hopefully you figured out that you have big P, and little p, two genotypes. So big P, little p is in 2 of the 4 squares, which equals 0.50, which equals 50%. Little p is in 2 of 4 squares, so same thing. Now, notice that you do not see a 2-to-2 genotypic ratio in the answers. That is because I simplified the ratio. So a 2-to-2 ratio is the same as a 1-to-1 ratio. All right, you take the smallest number in your ratio and divide it into the other numbers, right? So if you take 2-to-2, 2 divided by 2 is 1. So let me give you a different example of that. Let's see, how about a, this is not like a real genotypic ratio you'd see, but how about a 2-to-12 ratio? Here's my smallest number, I divide it into 12, so I get a 1-to-6 ratio. So hopefully that makes sense in terms of simplifying. Now, my other question that I would ask the class is, what kind of cross is this? So we have big T, little t, big V, little v, big T, little t, big V, little v. That is a dihybrid cross. Di means 2. We're looking at 2 genes, and you know that because there are 2 different types of letters and 2 alleles for each letter. Okay, so we would have a gene that we represent by T and a gene that is represented by V. So it's dihybrid. Gene 1 would have big T and little t. Gene 2 would have big V and little v. You know that's not all the same gene because I used 2 different letters. Remember, the alleles for one gene have to have the same letter. Okay. So that is it for looking at law of segregation, law of independent assortment, and Punnett squares. So the final lecture video for this module, actually I think I have 2 lecture videos, one on inherited diseases, and then I'm going to make a quick video on epigenetics.